for staying here because the room is almost as full as this morning, so it's really great to have you here. And when we were planning this event, we thought, so probably you're all very enthusiastic now to start epigenetics with your very organism, and we thought it would be good to give some guidelines how to do this. And the thing I would like to do, I would mention the RTP 3, which is Epigenetics in Ecology and Evolution, so that's a CNRS initiative, but many people are non-CNRS. So if you get more interested in this field, so I'd like to invite you to join this RTP and be part of the project. Okay, so when I started to write this cookbook for today, I was thinking, so what type of approach do I use? Do I use an approach with definitions? So, we have learned about all these difficulties to define epigenetics. And finally, I was very much inspired by a workshop Francesca just organized 1 week ago, and where was a person working on systems biology. Actually, a person, John Maynard Smith, probably, designed the idea that the epigenetic inheritance system is actually a part of the larger genetic system. So, obviously, this formula, that's the genotype, is a system with the environment will produce a phenotype. So let's say this was called genetic here or genotype for historical reason, and we would replace it by inheritance system. Well, in this case, we say this inheritance system is actually composed of a genetic part and an epigenetic part, and maybe others. It would interact with the environment and then produce a phenotype. So this is here a system, and this is a processus. And in this case, well, you can just pick, and we have to define the parts of this system and of this processes. So to define the components, and for the genetic system, of course, or genetic unit, it's a DNA, but can also be RNA. We have heard about viruses this morning. Epigenetics, so that would be the unit of DNA methylation, histones, non-coding RNA, topography of the nucleus, but also polycomb and trithorax proteins. So, depending on your biological system, the definition of the units will be different. It's interaction, so necessarily you have the time component, something we heard a lot also about. Then there's, of course, the environment, and we are here in the place where people know how to measure that environment. And finally, you produce a phenotype. Now, depending on what type of component you have in your system, you pick your method. Like if you study DNA, you can study it via heritability, for instance. Not Mendelian approaches, for instance, but you can also use molecular techniques, so exhaustive resequencing. The same thing for the epigenetic parts. You can study its heritability, or can even modelize its heritability, but you can study it also on a molecular level. Time. Well, time, then it's a unit to have to measure it. You can measure it in seconds and minutes and hours and generations. It can be, these generations can be separated by mitosis or by meiosis. Environment, it could be a stimulus, it could be a stress or a constant environment, and the phenotype could be molecular, so like measured with RNA seq, but it could also be non-molecular, something you observe or you have already observed. Now, I like this very much. The notion of time actually something that does not spring immediately into your mind is maybe that you even can use epigenetics to measure time. And this is just an example how the age of humpback whales was estimated using DNA methylation. So you imagine how difficult it is to sample humpback whales. Okay, so there's a relation of time and epigenetics. What I see as an advantage of this view, it's scalable, and I will come back to this later, so you can use different scales. It's not at all reviewer friendly because, to my knowledge, maybe I didn't dig deep enough, it was not proposed so far, so it's difficult to refer to something. Okay, scalability. Now, what you can now do is to pick your unit. Like most people will probably work on DNA, so that's the genetic part. That's fine. But it could also be RNA. And then pick your epigenetic unit. So it could be DNA methylation, it could be histone modifications, nuclear topography, non-histone proteins, etc. It depends on your biological system. And a lot of work will go into the definition of these units, because I trust you can measure time, I trust you can measure the environment, and I trust you can measure the phenotype. Now, let's say case one, you have no idea, no clue about how epigenetic system could look like in your specific biological model, plant, animal, or whatsoever. Now, Nicolas and I thought we give you some guidelines, really a cookbook, what you should do in these cases. Now, I will focus on DNA methylation and histone modifications, and I think Nicolas will go a little bit beyond this. Now, again, no idea. What you can measure is the degree of DNA methylation. You have some ELISA kits now, no association with Simon, but it could be another company. You measure with a very simple ELISA test or similar test the degree of methylation in your system. And sometimes, like Drosophila, well, there's some controversy, it's almost zero or very, very low. Well, you know this then. Or if you have a friend who has equipment, like chemist, you can digest DNA to mononucleotides and then run a LCMS or an HPLC. It gives you a measure. Something Christian will talk a little bit about it. You can also, you don't have to do any wet bench experiments. You can use the CpG observed expected ratio as an estimate to say how much DNA methylation you should expect in your system. And in this case, as I said, Christian will explain this in more detail. You have this distribution of CpG observed expected ratios. You have two peaks here, and what we know by experience now, this is the type of mosaic methylation, where some parts of the genome are methylated and other parts are not methylated. Well, histones then. How do you detect histones? Histones are proteins, and the easiest way to do it is to detect them by Western blot. It's trivial, but for many species, we have no idea if there are histones or not, actually. You can also digest the DNA, or more specifically the chromatin, with an enzyme that will cut in between the nucleosomes that are formed by these histones. And if there is a normal or expected chromatin structure, you will have this ladder. Each DNA ladder, every step of the ladder corresponds to the DNA that is wound around these nucleosomes. The enzyme cuts in between these nucleosomes, and you see here these different steps. Now, in the case where you have an unusual chromatin structure with nucleosomes that might not stick sufficiently well to the DNA, then you will have a smear. And that's actually very old methods to approach this idea. Old and not very costly methods, so I think we have to expect something like €1,000 to have this very first approach. And this is, I think, if you have no idea about your organisms, the first step to do. Now let's say you know a little bit. You know now that you have methylation of cytosine, and you know that you have histones, but you have no genome, which is well the case for most of our non-model organisms. I think there are currently two methods to address this question. One was recently published. It's called Genotyping by sequencing. It's a sort of extension of the earlier method that is called MSALP. So what you do, you use your DNA, you digest it with an enzyme that is not sensitive to DNA methylation. You fragment it, then you ligate adapters. You amplify, you ligate these adapters. You do a conversion step that will convert all unmethylated cytosines into uracil and subsequently in the amplification step to thymine, while all methylated cytosines remain intact. So in a way you fix this and you convert this DNA methylation difference into a sequence difference. And then you have a sequence difference and that you can sequence. So you send it out for linear sequencing. And the beauty of it is that if you have a reference genome, you can align these things to the reference genome and estimate your DNA methylation at these, around these restriction sites, so you will only sequence around the restriction sites. But you can also assemble by ultra-deep sequencing these regions around the restriction sites, and in addition estimate the DNA methylation there. So this method was developed for dandelions, so imagine there's no reference genome. Well, you can do the same thing, at least in theory, with DNase 1. So DNase 1, again, it's an enzyme that cuts in regions that are accessible, so the chromatin structure is more open. Very old method to detect these open region of the genome. You will cut it, again, ligate adapters, then pull down these things that has been digested and ligated to the adapters and sequence. So in principle, you can use the same method here, epigenotyping by sequencing for DNA methylation, but also for chromatin structure in organisms where there's no DNA methylation. And finally, case 3. So the estimated funding would say about 10,000 euro, so it starts to be a bit more expensive. And case 3, okay, you know there's 5-methylcytosine, you know about histones, and you have a genome, a reference genome. So in this case, you can do for the DNA methylation by sulfide sequencing, which was mentioned before, and it's exactly the same principle as I said before. You have unmethylated cytosine by treatment with this chemical compound by sulfide, it's converted into uracil. Amplification, it's thymine, so this is not methylated. If it's methylated, then it stays methylated cytosine, and finally in the PCR it's converted into cytosine. And this is something you don't have to do it in your lab. You just send out the DNA. You have to purify the DNA and you send it out. Either semi-public, so we work with Genotyping, we have worked with GATC, so these are two sequencing centers you can use. The GATC is slightly more expensive, 2 or 3 times. So you have this expertise outsourced somewhere. So what you have to do is just extract the DNA. Elena mentioned ChIP-seq. So as she said, essentially you pull down the DNA which is associated with a modified histone, which is part of the nucleosome, and then you get the DNA. Again, this is in general done. So this part of the project you have to do by yourself. This part of the project, sequencing, is done by a sequencing center. And then you have to, in general, again, do this part of the project, so aligning these bits of sequences to the genome to detect regions of enrichment of a specific histone modification, which is called a peak. Also for this part, so you will get the row reads, and you have to do something with these row reads. And so, as you might know, we are part of the CMAP, so we have a certain expertise in analyzing the data, and we supply this expertise, of course, to the members of the CMAP. We use an interface which is called Galaxy, and you as a CMAP member have access to this interface where this analysis can be done with a minimum of expertise necessary. Okay, so 3 different cases which might not correspond entirely to what you see as a specific problem with your organism, but I think it opens a perspective that epigenetics can be done, I would say, with any possible organism, and if you would like to integrate it in your studies, you should not be delayed by maybe the fear or the some worries about the technical parts. This is finally all very straightforward, and of course you have sometimes difficulties, but that can be solved. Now, very egoistic slide. We also are recruiting, so I think it's very encouraging to have seen in at least 2 slides now, or 3 slides now, these job offers. So that's an emerging area of research. And I give the slide to Nicolas.
Right, I will try to follow up on what Christoph has said, and I end my talk to the members of this LabEx seminar with some considerations, not so much guidelines, because I think everyone needs to set up their own, follow the technical guidelines, particular to certain techniques themselves, but more in terms of experimental designs before embarking into epigenetic study in a non-model organism. So the one thing I'd like to consider before doing a genetic study in a non-model organism is realizing this molecular framework that we've seen before and that I'm recapitulating here, because what it shows is that epigenome is not alone in this framework, right? So mostly in ecology and evolution, what we are interested in is variation, and most of us working in ecology and evolution, we study variations of phenotypes that we know very quite well. And if we work in non-model organisms, we are lucky if we have some genomes, and maybe we don't have all the rest of the components here. But it's true that phenotypic variations in the case of ecology and evolution will depend on the classical case of a mutant giving rise to mutant proteins that will affect the phenotypes. And that can also be contributed by modifications in gene expression, and this is where we are interested in with epigenetics mostly. And what makes things exciting in this field is that most of it is can be changed upon external conditions. And to make sure that you want to study this bit of the framework, I think it's important to also consider strengthening the molecular scaffold of your model organisms by describing a little bit more the other components of this. So what are these components? Christoph listed a few of them already. But the idea of these different modules, so genome variations, modification in gene expression, epigenome variations, and phenotypic variations need to be linked somehow in response to the environmental changes or the time scale differences that we're going to look at for our favorite organisms. So for example, for genome variations, I think it's good to have genome, but it must also be a little bit beyond that by comparing 2 genomes or 2 or 3 genomes, or even go to population genetics, which is nowadays a very powerful genetic technique to also find cis regulatory elements that could be that could explain evolutionary changes, for example. And this is absolutely something to consider as well. Modification of gene expression is an extension of your phenotype, and this is also something that needs to be described at some point if you want to link the epigenetic changes that you will observe in your phenotypic difference, because they will make this link to variation in gene expression. And in gene expression, here I'm talking about RNA, of course, which is the easiest way to study it in transcriptomic studies, but don't forget that the proteome component can also be sometimes uncorrelated to RNA due to post-transcriptional silencing, for example. So epigenome variations, there are several things that you can envision to study in epigenetic study for model organisms, and we've listed some of them. The main component that people study is, of course, chromatin, because this is a very well-known epigenomic feature for explaining differential gene expression. But there are also others like methylation, neuron topology, and so on. So the thing not to forget also is that very often we use epigenome as a proxy for annotating the non-coding part of the genome. I like to show this slide to my students because they sometimes don't realize that the coding part of a genome, this is here the human genome, and you all know, maybe you've seen this before, that the coding part is only 1.5% here, right? And if we go to epigenetics, what we're going to dive into is all of these unknowns. Some of them are functional, some of them are not, all right? You have a lot of transposable elements. Some of them will have an evolutionary effect, some of them will not. And you have some coding sequences, some non-coding sequences in there that will participate to gene regulation. And And the way to make sense of all this, what's happening, is that we use the epigenome as a mark, because we cannot make sense of what's in the genome besides coding regions based on DNA sequence alone very often. So we use epigenetic information to detect in there what are the functional regulatory elements of the genome. Several approaches do just that, like ChIP-seq, for example, or FAIR-seq or DNA-seq. They aim to discover regions that are promoters, enhancers, insulators, and other areas that could explain the changes in gene expression that you will have. And probably for non-model organisms, I would advocate that techniques like DNA-seq, FAIR-seq, MNI-seq, or ATAC-seq, something that's more based on the chromatin structure, will be sufficient for detecting most of these regulatory regions among the entire of the genome. Of course, you can be also you can also use some very well-known histone modification like H3K4ME3 and all the usual suspects for heterochromatin and euchromatin. But even in this case, you have to be careful, because sometimes these functions can be conserved or can be also evolved. And in non-model organisms, it's no surprise that you often find cases where these marks, this chromatin structure, don't even occur at all, or they even change functions in some cases. And this has to be documented way before embarking any cheap experiment in your exotic model, of course. So here an example is that the organism I study has very peculiar chromosomes where there's no centromere. So we have absolutely no idea how the heterochromatin in there will affect the expression of genes, because it's supposedly everywhere on the chromosomes. So supposedly there are changes in there, right? And I will conclude here by also talking a little bit about next generation sequencing, because mostly all that we've talked about in this day, and all that Christoph has talked about also, and myself, is now possible because of the emergence of next generation sequencing. And you've all seen this slide where in 2007 the introduction of these techniques in the field really decreased a lot the prices of genome sequencing, and the direct consequences was observed in the number of complete genomes deposited in NCBI, where from 2083 started to raise exponentially, because it was now possible to sequence, resequence, and perform additional sequence at the whole genome scale for any type of organisms that you want. Right? And that's actually what happened. Following this years, many different genomes for non-model organisms have been sequenced right after the integration of that. But there's a caveat, of course, and a very big difficulty for non-model organisms in embarking into NGS, as is the blocking of computing biology. And my final statement and consideration here will be that this is great because it has allowed many bioinformaticians to find job and matter to do, but there is a time now to reconsider what is relevant of bioinformatics and what is not. And for example, I would consider that just being able to align your FASTQ sequences onto a reference is not in the realm of bioinformaticians. Bioinformaticians, people who can reprogram, can really help us find new solutions to problems that haven't been solved before. But right now, and Christoph was saying that a bit with Galaxy, but I like to have this counterpoint with Christoph very often, is that we really need to take upon us, biologists, to use command line tools like Unix and R to use what's already been used for streamlined experiments. And I encourage everyone to at least train themselves, even by themselves, if that's possible, on these different tools to perform their projects. And with that, I think I fit in the 10 minutes, right?